Correct. So the current heart failure treatments in the clinic are mostly to relieve the symptoms, not to cure the underlying cause that leads to the heart failure. So basically, it's all about the loss of the muscles when you get your heart attack or MI, the massive loss of the cardiomyocyte or the muscle cells. And these muscle cells do not really regenerate. They cannot be replenished. So the heart becomes stiff and dysfunctional, and people go into the heart failure. The current strategies, though, best strategies would be to somehow replenish the loss of myocytes, make them new myocytes and new muscle cells in the heart that can restore the pumping function of the heart and elevates the symptoms of the disease. So what so the strategies, there are multiple strategies to that are currently being worked. One is you somehow reteach the muscle cells to proliferate or duplicate, which they lose this ability right after the birth, and they never replicate. And the other strategy is you give the stem cell derived muscle cells, you grow them outside the body, and you transplant those cells into the heart, giving them a new muscle patch, right? So this work is enhancing the endogenous repair mechanism, make the heart remember when it was proliferating, when it was in the developmental stage or in utero, and what it lost the ability to basically proliferate again. So what we did was that the PSAT one is a gene that is a multifunctional gene. If you look for the genes that are expressed embryonically when the heart muscle cells are proliferating and growing, and the ones that are lost after the birth, and also the genes that are involved in multiple aspects of the heart. So heart failure, okay, you do lose the muscle cells, but there's also a lot of other things going on, like reactive oxygen species, inflammation, loss of vasculature. So a gene that can affect the multiple aspects of the disease heart might be more productive than a single gene or single functional gene therapy kind of stuff. So that's how we came up with this PSAT1, which is involved in generating amino acids called serine and glycine. And these are very important nucleic acids to involve in multiple functions by reducing the reactive oxygen species, by creating purines that leads to the DNA synthesis in the muscle cells and make them duplicate. So clinically, we use it, application would be, if it pans out, is we use a technology called modified RNA. And modified RNAs are base therapies, not only for heart, for other ailments too, are now a hot topic these days after the COVID vaccine was made by the modified RNA techniques for the Nobel Prize on that. The good thing about modified RNAs, rather than giving a whole DNA as a gene therapy, which may integrate into the heart itself, this modified RNA doesn't integrate inside the genome. It very quickly translated, makes the protein very fast, and it's transient and goes away. And it is also protective against the RNases that are in the heart, rather than naked RNA. So the implication would be to use this data that we generated in the mouse model, test them in the larger animal model system, which are more closer to human. And if it pans out, maybe one day it could be a therapeutic to be given to the patient just after the MI to induce the cardiac proliferation and relieve the symptoms of the heart attack and maybe prevent them going into heart failure. So in our study in the mouse model, we found that there's a substantial reduction. It does two things in the context of myocytes. The reduction in apoptosis or the cell death of the myocytes is about 15% compared to control mouse. So it's 15% more muscle cells survive when you treat this with this particular modified RNA gene. And the second aspect is about 8% renewal. So myocytes begin to divide. So in sum total, both preventing the myocyte from death and regenerating the 8% of the myocyte. It may not sound great, but it's a one-dose treatment. And if you can induce 8% of the myocyte renewal, that could be substantial. The benefit is using this technology, as I was talking to you, is a very pulse-like, very short-lived, transient expression of the protein required. So protein is made, it does its job. Now the cells react to the new serine that are found by this gene, and this gene goes away with the modified RNA. It doesn't integrate. There's no long-term expression of this gene. It's the short-term pulse-like protein activation. So it's transient reactivation of embryonic gene rather than a permanent activation of embryonic gene. You're right. Permanent induction of embryonic gene might have safety concerns, tumor formation and things like that. So that's why we deliberately use a technique that is going to give this embryonic gene to the adult heart short term, 7 to 8 days. And the downstream effect of this re-expression of the gene is what the cells respond to. There are already clinical trials using the modified RNA platforms are going on. There's a whole company called Moderna. The whole product is modified RNA-based technology. The translation technically would not be challenging, but the safety issues and the functional aspects of this particular gene needs to be verified into the larger animals like pig and sheep and dog, which are more closer in anatomy and physiology to the human beings rather than the mouse. That's one aspect of it. Other aspects of it, the way the practical is that when the patient have heart attack, they come to the clinic to get their arteries open. The gene could be delivered at the same time when the patient's in the hospital into the heart. So it does not be technically challenging to give this particular gene modified RNA into the heart. I can foresee that. Intervention is early, so it prevents cell death and reactivate the myocyte proliferation, leading to a better outcome after MI, and maybe prevention of going a patient into the heart failure. Now, one drawback of our technology is that we are not targeting a single cell type in the heart. So when you give the modified RNA, it will go to cardiomyocytes as well as to the other cells in the heart, right? There are technologies that we are developing and other people are developing where you can target a specific cell. You use the promoter of cell, a specific promoter, to target your delivery of this modified RNA to a desired cell type, not to the all cell type. So that might be one challenge. But on the other hand, because it is a pleiotropic or multifunctional gene, and we showed that it leads to the not only cardiomyocyte effects, but also the effect on the vasculature. There are more vessels, and there's less reactive oxygen species, less DNA damage. So I think it might be beneficial not to give only to the myocytes, but rather to the heart. So again, heart can respond, and the whole environment of the heart is better suited now for the myocytes. Once they are regenerated, they can survive. Because if the reactive oxygen species and the DNA damage elements and no vasculature are there, even the new myocytes will not get fed by the new vasculature, and they may eventually die. So I think the probably giving it to the heart rather than to just the cell type might be a better approach. There has to be safety issues. We apparently we look at the safety aspects of it, but not very closely. That was not the goal of the study. So obviously the safety profile has to be whether a single dose is enough or you need a multiple doses to create an environment that lasts longer beyond beyond just the select window, the long-term goal. And then obviously, mice and humans share a lot of things, but they're not exactly the same physiologically. And so validation of these studies into the large animal model systems might be a prerequisite before this technology can move into the real human patients.